Hello everyone. In this video we want to make an experiment. This experiment to show you the phases of mitosis. You know, there are four phases of mitosis. At the end it will give two identical cells. They have the same genetic material. In this experiment we're going to use garlic. Actually, you can use also onion. Here we will use the root tips of garlic to study the mitosis process. You know, when the roots start to grow, they are doing what? Which type of cell division? Which is what? Mitosis, right? To give the same type of cells. Here they should undergo what? Mitosis division, because it's growing. Okay, so here you have to make like a scientific report. In the scientific report you have to just hold on. You have to write the title, then you have to write what is the goal, then you have to write the materials and tools. Listen carefully, they are required from you for the first exam. Also the procedure, the steps, step by step. Okay, what did we do, and your observation. In your observation you have to draw the cells and what can you see under the microscope. In the materials and tools you have to know what is the purpose from using each of the materials they are mentioned. And for what? Maybe I'll ask you in the exam, this solution it's for what? This tool it's for what? Okay, you have to write what is the purpose of each one of them. For sure we're not putting something like just to write materials or tools. There is a purpose from each one of them, right? So let's start.
In this video, we'll be preparing root tips of allium species to observe cells in various stages of mitotic division. Mitosis is one of the stages in the cell cycle where the chromosome of a cell replicate and separate to eventually form two genetically identical cells. Unlike in meiosis, chromosome number is maintained in both daughter cells. For the experiment, you will need onion or garlic bulb, beakers, toothpicks, carnoise fluid.
Onion, beakers, carnoise fixative solution. This one is very important. Now we will talk about it. Why do we need it? While we are doing the experiment, I'll tell you why do we need this one. Okay, continue.
70% ethanol.
This ethanol also, it has a purpose.
1 normal hydrochloric
acid.
Acetocarmine or
acetocarmine. This one is the stain or the dye. We use it to stain the cells. Okay.
Acetoarsine stain. Glass slides and cover slips. Blade or scalpel. Watch glasses or petri dishes. Tiny vials or containers. Spirit lamp or.
This one here, we need a source of heat. It doesn't matter. You can write Bunsen burner, any source of heat. Okay.
Bunsen burner or any flame source. Blotting paper.
Here you can use a blotting paper or you can use a filtered paper.
Droppers. Thumb forceps and a pair of scissors. Compound light microscope.
So repeat the video to write all these tools required. Let's continue.
And immersion oil.
This is the last one, immersion oil. We will talk about it. Why do we need it? How to use it in the microscope when you use the high power objective lens. Listen now.
To begin the experiment, take an onion or garlic bulb and fix it on a beaker containing tap water using toothpicks like you see here. Make sure the base of the bulb touches the water level.
Look again. This is the garlic. Okay, put it the bottom. It should be faces what? The water, to allow the roots to start to grow here. Okay.
Keep this in a safe corner for a couple of days. Once the roots have grown about 2 to 3 centimeters, cut out 1 centimeter of root tips and transfer them into a tiny vial containing carnoise fixative fluid.
Listen, you cut it now. And you put it in a container. This container, it contains what? Carnoise fluid. or a fixative solution. Why? What is the purpose of this fluid? Actually, this one it will fix the DNA for the root tip cells. Okay, fix the DNA for the root tip cells. Listen now again.
Corner for a couple of days. Once the roots have grown about 2 to 3 centimeters, cut out 1 centimeter of root tips and transfer them into a tiny vial containing carnoise fixative fluid. Carnoise fluid fixes the DNA of the root tip cells. Leave the roots in the fixative for about 48 hours. If you're planning on storing the root tips for a longer time for use in future, you may transfer the fixed root tips in a second vial containing 70.
Listen. If you want to use this in the future, you can, after you put this root in the fixative solution or carnoise, actually, you can transfer them into 70% concentration of what? Hydrochloric acid. Okay?
If you're planning on storing the root tips for a longer time for use in future, you may transfer the fixed root tips in a second vial containing 70% ethanol. Ethanol dehydrates the root tissue and thereby preserves DNA. Root tips preserved in ethanol and preferably refrigerated will keep well for a couple of years for genetic studies. Either ways, take a few root tips from carnoise fluid or ethanol and transfer them onto a watch glass containing 1 normal hydrochloric acid.
Okay, so here he has mentioned about the ethanol, so to dehydrate the root tissue and preserve the DNA. You can put it in the refrigerator for for a year, for example. Okay, then wherever you put it, in the carnoise fluid or in the ethanol, when you want to start to make the experiment, you have to put it in a dish. This dish, it has inside it what? It has inside it hydrochloric acid, which is HCl. Okay, it has here hydrochloric acid. Actually, what is the purpose of the hydrochloric acid? Actually, it softens the cell wall. You know, you're talking about a plant cell, so the hydrochloric acid, it will what? Soften the cell wall. Listen.
Acid will soften the cell walls and weaken cellular connections, so it becomes easy to squash the root material later. Gently warm the watch glass on a flame for about 5 seconds. Expose the root tips in the acid for about 2 minutes. Give the root tips a couple of washings in distilled water.
Wash 2 times. You have to wash it in distilled water, as you see.
Now transfer the roots onto a watch glass containing acidocarmine or acido-orcine stain. These are excellent chromosomal stains and will impart a deep red coloration to the nuclear material of the root cells.
It's very important to know
Warm
this.
the stain on the flame for about 5 seconds and leave the root tips in the stain for about 5 to 10 minutes. Now transfer the root tips onto a clean glass slide containing a drop of water. Using a sharp blade or scalpel, remove about 1 millimeter of the root tip and discard the rest.
I need the root
The very
tip
tips
only.
of the roots are the regions with active cell division, and they are all that we require for a mitotic experiment.
So for the mitotic experiment, I need only the tips, okay? And the rest, I'll remove it. I don't need it, because where the tips, where they are starting to making the mitotic, they are dividing, okay? So here it's easily to see the mitosis phases in these cells, okay?
Gently lower a cover slip on the root tips, making sure to avoid any air bubbles. Using the blunt end of a forceps or a pencil, gently tap the cover slip a few times until the root tips are uniformly squashed in between the slide and the cover slip. A properly squashed slide will appear faint cloudy pink to almost colorless. The root tip slide is now ready for microscopic observation. This is how a finely squashed root tip appears under 10x of the microscope.
So in the microscope, on the objective lens, which is 10x, this is what you're going to see. Okay, you have to draw this. This is the first observation on 10x objective lens. That's what you're going to see. Don't forget, here you are seeing what? Plant cells. These plant cells, they have a regular shape. Listen now.
As you can see, the cells of the root tips are nicely spread out with almost no overlappings. If root tips are not squashed enough, you may repeat the squashing process until you get well spread root cells. Scan the slide for cells showing mitotic division stages under low power and then proceed to observation under high power, such as 40x or 100x oil immersion lens. For observations under 100x objective, you will need to add a drop of immersion oil on top of the cover slip and gently lower the objective lens until it touches the oil surface.
So here, look, where did he add the immersion oil? He put it on the cover slip, not on the sample. Then he started to make this up to touch the lens to see this one in 100x. Listen now.
From there, use the fine adjustment knob for focusing. You may take micro photographs of the various stages by employing a simple technique using a mobile phone and a tripod, like so. And here's a quick slideshow of the various mitotic stages of cell division in root tip cells of Allium species.
So look at the first one, which is the interface. This is the interface. This is the resting stage, and this is in most of the cell life. Listen now. Now this is in the prophase. Prophase here, actually early at the beginning of the prophase, here chromosomal condensation initiates, giving the nucleus like grainy, as you see, like has grains inside its appearance. The nucleus is still visible, still, till this time the nucleus or the nuclear membrane still appears. You have to draw all of these, guys. This is now for the prophase late. Look how they start to break the nuclear membrane. Further chromosomal condensation results in the thick rope-like chromosome. They start to make like X-shaped structure, actually, which begins to assemble into chromatid and centromere. This one is metaphase. As you see, this one here, this one cell, and this is another cell. Here they are granules, which is a prophase, and this here in this cell here, they are starting to making the metaphase. As you see in the metaphase here, the cell they are at the middle of what, of the cell, as you see. Okay. This is early anaphase. Look at the anaphase, how the chromosomes start to go to the other side, like this one, this one, this one here. They are connected. This is the centrosome, sorry. Look, and this one here. Can you see? Draw them. Okay, continue. This anaphase here, sister chromatid, reach the opposite. Look how they are far away now. They are to the opposite pole in the equal number and in equal number, as you see. This is the early telophase. Look at the early telophase here, how start a nuclear membrane start to be formed, which is here and here. As you see here, telophase also. Here chromosome almost fully condensed. Start also the chromosomes in the telophase when they go to the opposite side and decide to make in a new nucleus, start to less condense, okay, to form original chromatin network, to go back again as spaghetti, okay. Here in this one, as you see, if you remember, we said for the plant cells they are making what cell plate. Here the late telophase, the early cytokinesis, the cell plate start to be grow, which is here. A chromosome individually is lost, forming what? Chromatin. Now start to make like a chromatin, like how they are here, and start to make like a chromatin again. Okay, not X shape, but I'm doing more than one. Okay, they are look like spaghetti, only thread-like structure. Okay, nuclear membrane and the nucleolus appear, the cell plate begins to form, and here to start what? The division of the cytoplasm. So I hope this one it was helpful to understand the mitosis. If you still have a question about it, you can send me your question. Don't forget to send me what? The lab report. Thank you for listening.